Hi, my name is Damián, and today I'm going to talk to you about how leaf-litter weevil richness increases with altitude in a tropical temperate transitional forest in northeastern Mexico. Altitudinal variation in communities' species richness has been amply studied in the past. McCain in 2009 summarized it in 4 common patterns: decreasing species richness as elevation increases, a richness plateau at lower elevations, the same plateau at lower elevations with a mid-elevational peak, and a mid-elevational peak throughout the altitudinal gradient. In the Mexican mountains, several variation community studies across elevational gradients have been performed. In a recent 2021 study carried out in the highest Mexican mountain, a decreasing mid-elevation peak richness pattern was found for dung beetles, Scarabaeidae. There has been a bias towards Scarabaeidae while exploring the diversity patterns of coleopterans in Mexican mountains, while other groups have gone underrepresented, such as Curculionidae. Leaf-litter weevils are of special interest because they have high rates of diversity, dissimilarity, and endemism worldwide. That is why the objective of this project was to determine the diversity pattern of leaf-litter Curculionidae in a tropical temperate transitional forest along an elevational gradient. We expected that tropical weevils would be dominant at low elevations, while temperate weevils would be dominant at higher elevations. We also expected that as elevation increased, weevil species richness would decrease. El Cielo Biosphere Reserve is the northernmost tropical temperate transitional forest in the Americas, located in the state of Tamaulipas, Mexico. It is located in the eastern Mexican mountain range, known as Sierra Madre Oriental. Notice how the sierra is not further connected up northeast. Leaf-litter samples were collected along an elevational gradient from 200 to 2,000 meters above sea level. All the elevation gradient was primary forest. 2 plots per elevation floor were sampled for a total of 20 plots. Within each plot, an approximate of 15 1-meter square leaf-litter samples were processed, giving nearly a total of 300 processed leaf-litter samples. Adult weevils were separated into morphospecies by visual inspection, and at least 1 weevil per morphospecies per plot was mounted and labeled. Weevils were identified by expert taxonomists, such as Manuel Barrios, Robert Jones, Lourdes Chamorro, Bob Anderson, and Francisco Armendariz. 81 weevils morphospecies were separated. 56 were labeled as leaf-litter species. According to expert opinion, the remaining 25 were excluded because they don't occur in leaf litter. They live in herbaceous vegetation, shrubs, or the canopy. We also excluded Scolipini. 291 samples were processed. According to the Chow-2 richness estimator, we observed 76% of the total leaf litter weevil species, while according to the Jackknife 1 estimator, we collected a total of 82% of the predicted richness. The number of predicted species increased at a rate similar to the actual randomized species accumulation curve. Accumulation curves of weevil species for each of the locations show that the observed total species richness ranged from 4 to 9 at each elevation of floor. Coverage-based rarefaction curves show that most elevation floors were sampled near inventory completeness, except for the 800-meter plots. We believe this was due to a methodological artifact. Sample completeness rarefaction curves with respect to sample size also tended towards inventory completeness for each elevation floor. The most abundant weevil genus was Europtus with 7 morphospecies, followed by Trachyphlomimus, Dioptrophorus, a new genus of Cryptorhynchini, currently being described by Manuel Barrios, Theognete, and Anchonus. Our most surprising result was to find an increase in leaf-litter weevil species and abundance as elevation increased. The relationship between richness and elevation was better explained by a quadratic model showing a mid-peak pattern around 1,600 meters. Although this may change in the future because the tendency may be better explained by a non-linear model. The pattern is opposite to the one found for ants sampled in the same plots of El Cielo in a parallel study carried out by a colleague. El Cielo reaches a maximum elevation of 2,300 meters above sea level, but the majority of the highest parts are located between 1,900 and 2,000 meters, which means that in our study we cover the entirety of the mountain and do not have a truncated gradient. El Cielo elevation gradient ends in forest, while other mountains end in alpine grasslands. The presence of forest at the highest elevations could be mitigating the decrease of forest limb diversity, such as leaf-litter weevils. The presence of a primary forest spanning the entire elevational gradient from 200 to 2,000 meters of altitude could be a driving factor of the Weevil richness pattern. The best fit generalized linear model for Weevil richness showed that elevation explained over 60% of the total variation, while the interaction between DBH and precipitation seasonality explained over 20% of Weevil species richness variation. A canonical correspondent analysis explained over 30% variance in the composition of the Weevil communities of El Cielo. CCA axis 1 mostly represented elevation, because lower elevational floors and lowland Weevil species were clustered near the left, while higher elevational floors and highland Weevil species were clustered near the right. For the case of the most abundant Weevil, Europtus, the genus identity did not predict the ecological behavior of the morphospecies, as these were widespread across the CCA. As a contrasting example, the Trachyphlomymus weevils were clustered near elevational floors dominated by oaks. Leaf-litter weevil species, the similarity was high along El Cielo elevational gradient. Incidence-based indices such as Jacquard and Sorensen and abundance-based index, Bray-Curtis, show that the similarity of 87, 78, and 93% respectively. All indices led to the same pattern. Turnover explained more dissimilarity than nestedness. The highest differences in Weevil species and individuals' abundance occurred between the base and the top of El Cielo elevational gradient. We could not reconstruct the biogeographic origin of a Weevil taxa, since Curculionidae is largely understudied and lacks species and genus level phylogenies for most neotropical communities. But we tried to reconstruct the biogeographic affinities using contemporary distributions and expert taxonomists' opinions. We chose to classify the affinities as neotropical or nearctic. Only 3 out of the 25 leaf-litter Weevil genera sampled in our study had a distribution which made us label them as nearctic. These were genus Canistes, Trachyphlomymus, and Cosonus, although it is worth mentioning that all of these labels are tentative. Regarding Weevil morphospecies, out of the 56, only 5 were nearctic and 51 were tropical. And well, furthermore, because El Cielo is located at the Sierra Madre Oriental and the Sierra is not further connected up northeast, colonization from southern tropical lineages could have been more likely than colonization of northern temperate species. But before any conclusion is reached regarding this topic, phylogenetic analyses are needed to infer biogeographical origin. Summarizing, in El Cielo Biosphere Reserve, leaf-litter Weevil species richness peaks at 1,600 meters of altitude. Although most Weevil lineages are tropical, their richness and abundance increase with elevation. The main predictor of Weevil community variation was elevation, followed by the interaction between tree size and precipitation seasonality. As precipitation increased along the gradient, Weevils preferred bigger trees. And the similarity across the Weevil community is mostly caused by turnover. Communities at 200 to 2,000 meters, 200 to 1,000 meters of altitude are more similar than those between 1,000 to 2,000 meters of altitude. I'm thankful for the support of all the people that made possible the realization of this project, and thanks to everyone that listened.